Hello बच्चों, welcome to Hello Biology. This is the 8th lecture of chapter Principles of Inheritance and Variations. Students, till now we have understood incomplete dominance, where neither allele is completely dominant, and the heterozygous individual shows an intermediate phenotype. Now, let us move to another important exception of Mendel's law of dominance, codominance. Let us now understand this fascinating inheritance pattern in detail. When two different alleles are present together in a heterozygous individual, both alleles express themselves fully and simultaneously. Neither allele dominates the other, and neither allele gets masked. Also, unlike incomplete dominance, no intermediate phenotype is formed. Instead, both parental traits appear together in the same individual. That is why such alleles are called codominant alleles. Let us understand this with some examples. In the ABO blood group system, the alleles IA and IB are codominant. When both are present together, the individual has AB blood group and expresses both A and B antigens. Another example is Roan cattle, where both red and white hair are present together on the body. The offspring is neither completely red nor completely white. Similarly, in speckled chickens, both black and white feathers are visible simultaneously. So students, remember, in incomplete dominance, an intermediate phenotype is formed. In codominance, both traits are expressed together and independently. This complete and simultaneous expression of both alleles in a heterozygous individual is known as codominance. Codominance can be observed in several organisms, but one of the most important and commonly studied examples is the ABO blood group system in humans. Now that we have understood the basic concept of codominance, let us explore how both alleles express themselves simultaneously through the fascinating example of ABO blood groups. Codominance in humans, the ABO blood group system. First of all, notice the statement at the top of the slide. The ABO blood group system is controlled by 3 alleles, IA, IB, and I. Since more than 2 alleles are present for the same gene, this is also an example of multiple alleles. Now let's understand each blood group one by one. First, blood group A. A person can have either genotype IAIA or IAI. The red blood cells of these individuals contain A antigens on their surface, and their plasma contains anti-B antibodies. Next, blood group B. The genotype can be IBIB or IBI. These individuals have B antigens on their red blood cells and anti-A antibodies in their plasma. Now, look at blood group AB. Its genotype is IAIB. This is the most important blood group for understanding codominance. Here, neither IA nor IB dominates the other. Instead, both alleles express themselves completely and simultaneously. As a result, both A and B antigens are present on the surface of the red blood cells. This is why blood group AB is considered the classic example of codominance. Finally, blood group O. Its genotype is II. Since the recessive allele I does not produce any antigen, no antigen is present on the red blood cells. However, both anti-A and anti-B antibodies are present in the plasma. So, students, the most important point to remember from this slide is IA and IB are codominant alleles. When both are present together in genotype IAIB, both A and B antigens are expressed equally. Therefore, blood group AB is the best example of codominance in humans. This shows that in codominance, both alleles express themselves fully without masking each other. Students, before we understand how ABO blood group is an example of codominance, let us first understand the dominant-recessive relationship present in this system. As we discussed earlier, the ABO blood group system is controlled by 3 alleles, IA, IB, and I. Now let us focus on the relationship between IA and I. When an individual has the genotype IAI, both alleles are present together, but they do not express equally. In this case, IA behaves as the dominant allele, while I behaves as the recessive allele. Therefore, only the effect of IA is visible. As a result, A antigen is produced on the surface of the red blood cells, and the individual shows blood group A. The recessive allele I remains present in the genotype, but does not express itself in the phenotype. So, students, this is a simple example of a dominant-recessive relationship, where one allele masks the effect of the other. IA equals dominant allele, I equals recessive allele, genotype, IAI, phenotype, blood group A. Students, we have just understood the dominant-recessive relationship between IA and I. We learned that when these two alleles are present together, IA dominates over I, and only the A antigen is expressed. As a result, the individual shows blood group A. Now, let us look at another dominant recessive relationship in the ABO blood group system, the relationship between IB and I. Just like IA, the allele IB is also dominant over the recessive allele I. Therefore, when an individual has the genotype IBI, the dominant allele IB expresses itself and produces B antigen on the surface of the red blood cells. The recessive allele I remains present in the genotype but does not show any expression. As a result, the phenotype of the individual becomes blood group B. So, students, remember, IB equals dominant allele. I equals recessive allele. Genotype equals IBI. Phenotype equals blood group B. Till now, we have seen two dominant recessive relationships in the ABO blood group system. IA dominates over I. IB dominates over I. But now comes the most interesting question. What will happen if IA and IB come together in the same individual? Will one dominate over the other? Or will something entirely different happen? Let us find out, because this is where the concept of codominance begins. Students, till now we have understood two important relationships in the ABO blood group system. First, we saw that IA is dominant over I, producing blood group A. Then we learned that IB is dominant over I, producing blood group B. Now comes the most important and interesting question. What happens when IA and IB come together in the same individual? Will IA dominate over IB, or will IB dominate over IA? The answer is neither. When both IA and IB are present together, neither allele behaves as dominant or recessive. Instead, both alleles express themselves completely and simultaneously. This phenomenon is known as codominance. Let's look at the slide carefully. On the left side, we can see two alleles, IA allele, which produces A antigen, and IB allele, which produces B antigen. When these two alleles come together, the genotype becomes IAIB. Now, instead of one allele masking the other, both alleles remain active. As a result, both A antigen and B antigen are produced. On the right side of the slide, you can see a red blood cell. Notice that both A antigens and B antigens are present on its surface. This is the direct evidence that both alleles are expressing themselves equally. Because both antigens are present, the phenotype becomes blood group AB. Now look at another interesting point. Individuals with blood group A have anti-B antibodies. Individuals with blood group B have anti-A antibodies. But individuals with blood group AB have neither anti-A nor anti-B antibodies in their plasma. Why? Because their body recognizes both A and B antigens as its own. Therefore, it does not produce antibodies against either of them. Students, this is why blood group AB is called the universal recipient, because it can receive blood from all ABO blood groups. Now let's connect this example with the definition of codominance. In codominance, both alleles are present together. Neither allele suppresses the other. Both alleles express themselves fully. Both traits appear simultaneously in the phenotype. And that is exactly what we observe in blood group AB. So students, remember this important conclusion. Genotype, IAIB. Both IA and IB are expressed equally. Both A and B antigens appear on RBCs. Phenotype, blood group AB. This makes the ABO blood group system one of the best and most famous examples of codominance in humans. Let us look at another classic example of codominance, Roan coat color in cattle. In cattle, coat color is controlled by two alleles, CR for red coat color, CW for white coat color. First, look at the cow on the left side. Its genotype is CRCR. Since only the red allele is present, the cow produces only red hair and appears completely red. Now look at the cow on the right side. Its genotype is CWCW. Since only the white allele is present, the cow produces only white hair and appears completely white. The most interesting case is the cow in the middle. Its genotype is CRCW. Now, students, think for a moment. If this were a case of complete dominance, either the red color or the white color would completely mask the other. If it were incomplete dominance, we would expect a completely new intermediate color. But that is not what happens. Instead, both alleles express themselves fully and simultaneously. As a result, the animal produces both red hairs and white hairs at the same time. When we observe the coat, we can clearly see a mixture of red and white hairs throughout the body. This unique coat color is called Roan. Notice carefully that the cow is not pink or any intermediate color. The red hairs remain red and the white hairs remain white. Both colors are visible together. This is the hallmark of codominance. So, students, remember, CRCR equals red cow, CWW equals white cow, CRCW equals roan cow. And in the roan cow, both alleles are expressed equally without masking each other. Therefore, roan coat color in cattle is one of the best examples of codominance, where both parental traits appear together in the heterozygous condition.
That's all for today's lecture, students. Thank you for watching. Don't forget to subscribe, like, comment, and share. See you in the next lecture. Goodbye and happy learning.